Welcome to another video of the AS Biology series. To those of you all doing paper 1-1, this one is for you. This is the 9700 May-June 2019 paper 11. If you are new to this channel, do support the DF Edu Squad by becoming part of it. And all you have to do is just hit the subscribe button. It's that simple. Also do like and share with your friends too. Then let's just start straight away. Question 1. A student was told that the actual length of a cell structure is 5 micrometer. The student was asked to state an equation that can be used to calculate the magnification of an electron micrograph of their cell structure. The student used some of the letters Q to U in the equation. Q is equal to the length of the cell structure image on the micrograph in centimeters. R is equal to the length of the cell structure image on the micrograph in millimeters. S is equal to 1000. T is equal to 1 upon 5, U is equal to 5, which is the correct equation to calculate the magnification. Now magnification is nothing but image size upon actual size. Therefore, the image size is equal to R times S. As R is measured in millimeters and R is the image size, we have to convert it into micrometers. Therefore, in order to convert it into micrometers, we have to multiply by 1000. So R times S will give you the image size in micrometers, then that answer divided by the actual size, which is 5 micrometer, will give you the magnification. In other words, it is R times S times T. Hence the answer is D. Question 2. Which features of cilia and root hairs are correct? The columns are increased cell surface area, cannot be resolved with the light microscope, contain vacuoles, more than one present on a cell. Now vacuoles are only present in plant cells, hence in root hairs. Therefore, we can rule out options B and D, leaving us with the answers being either A or C. In one root cell, there is only one root hair, whereas in one ciliated cell, there can be many cilia. Hence, the answer is C. Increased cell surface area, root hairs, cannot be resolved with the light microscope, cilia, contain vacuoles, root hairs, more than one present on a cell is cilia and not root hairs. Question 3. Which are functions of microtubules? 1. Allowing movement of cilia in a bronchus. 2. Attachment of centromeres during metaphase. 3. Moving secretory vesicles around a cell. Now the functions of microtubules are, they participate in the formation of the spindle, they are major components of cilia and flagella, they assist in the movement of organelles as well as vesicles. Therefore, if we go through the options 1, 2 and 3, actually all the options are functions of microtubules. They allow the movement of cilia, they also form the spindle, hence the attachment of centromeres during metaphase, as well as moving secretory vesicles around a cell. Therefore, the answer is A, 1, 2 and 3. Question 4. Some cell structures are listed. 1. Mitochondria, 2. Nucleus, 3. Chloroplasts, 4. Ribosomes. What is the correct order of these cell structures when listed from largest to smallest? Now, one thing you should know is that the smallest cell structure is the ribosome and the largest cell structure is the nucleus. Therefore, if we go through the answers A, B, C and D, the largest being the nucleus and the smallest being the ribosome, that leaves us with the answer being B. 2 followed by 3, then followed by 1 and lastly 4. Question 5. Which cell structures have ribosomal RNA? 1. Chloroplast, 2. Mitochondrion, 3. Nucleus, 4. Rough endoplasmic reticulum. Since all 4 structures have ribosomes either being 70S or 80S, they will all have ribosomal RNA. Therefore, the answer is A, 1, 2, 3 and 4. Question 6. A cell structure in the macrophage destroys bacteria. Some bacteria stop this cell structure from functioning. Which cell structure in the macrophage is stopped from functioning by the bacteria? Now, when a macrophage engulfs bacteria, a phagosome forms, which is a vesicle containing the bacteria. Then digestive enzymes are secreted by lysosomes which attach to the phagosome, which helps to digest the bacteria. In order to stop the cell structure in the macrophage destroying bacteria, lysosomes should be inhibited. Therefore, the answer is B, lysosome. Question 7. A solution of amylase was added to a suspension of starch. After 30 seconds, 3 samples of the mixture were tested with iodine solution, Benedict solution or with biuret reagent. Which are the expected results? The color with biuret reagent should be purple as amylase is an enzyme and all enzymes are proteins. Therefore, the answers can be either A or C. And since the function of amylase is to break down starch to simple reducing sugars, there should be a positive test with Benedict solution. Therefore, the answer can't be C, leaving us with the answer being A. Iodine solution black, Benedict solution green, Biuret reagent purple. Question 8. A student carried out a Benedict's test on several different known concentrations of alpha glucose. Which graph represents the results correctly? Higher the concentration of reducing sugar, faster the color change. Faster the color change, less time taken. Hence, as the concentration of alpha glucose increases, the time taken to first color change should decrease with increase in concentration of alpha glucose. Therefore, the answer is A. Why cannot the answer be B? It is because they have plotted concentration of alpha glucose on the y-axis and time to first color change on the x-axis. However, the time depends on the concentration and the dependent variable usually goes on the y-axis and the independent variable on the x-axis. Therefore, since the concentration is known, it should be on the x-axis. That leaves us with the answer being A. Question 9. The diagram shows 3 hexose sugars. Which row correctly shows examples of carbohydrates in which these 3 hexose sugars occur? Now I have labeled under 1, 2 and 3 what each of them are. So 1 is alpha glucose, 2 is beta glucose, 3 is fructose. How do we know which one is alpha and which one is beta? We have to consider the OH group on the first carbon atom that lies on the right side after the oxygen atom. If the OH group lies below the plane, then that is alpha glucose. If the OH group lies above the plane, then that is beta glucose. And cellulose is made up of beta glucose. Therefore, cellulose should contain number 2. Starch is a polymer of alpha glucose. Therefore, should contain 1. And sucrose is made up of glucose and fructose. And since 3 is fructose, 3 belongs to sucrose. And that leaves us with the answer being D. Question 10. What can occur during condensation of carbohydrates? A. A disaccharide is produced from monosaccharides. B. Glycosidic bonds are broken. C. Molecules of water are used up. D. Monosaccharides are produced. Now, condensation reactions are where bonds are formed, whereas in hydrolysis, bonds are broken. Condensation forms water, whereas hydrolysis uses water. Since they are asking about condensation, answer C is incorrect. Molecules of water are not used up. Answer D, monosaccharides are produced. That is also incorrect, as condensation is forming bonds and not breaking bonds. B, glycosidic bonds are broken. That is also incorrect. A disaccharide is produced from monosaccharides. Yes, as when monosaccharides join together, they form bonds. Hence, condensation is involved. Therefore, the answer is A. Question 11. What is true about triglycerides? The columns are hydrophobic and insoluble in alcohol. Triglycerides are soluble in alcohol and are hydrophobic in nature. Therefore, there should be a cross mark in insoluble in alcohol and a tick in hydrophobic. And that leaves us with the answer being B. Question 12. What is the minimum number of carbon atoms in an amino acid? Each amino acid has a COOH group as well as 2 NH2 groups. Therefore, requires minimum 2 carbon atoms, not 1. Hence, the answer is B. Question 13. In an investigation, the same concentration of the enzyme phosphorylase was added to different concentrations of glucose phosphate and incubated at 30 degrees Celsius. At 1 minute intervals, 1 drop of the reaction mixture was removed and added to a drop of iodine solution on a white tile. The diagram shows the results of this investigation. What explains the trend in the results of this investigation? In a rate of reaction, initially the substrate concentration is limiting, and towards the end, the enzyme concentration becomes limiting. If we go through the answers A, B, C and D, A. Phosphorylase catalyzes a reaction converting glucose phosphate to starch. Yes, that statement is true, but that doesn't explain the trend based on the results of this investigation. Hence, answer A is incorrect. B. The maximum rate of reaction is reached at 20 mg per dm cube of glucose phosphate. That is incorrect, as maximum rate of reaction is reached at 30 to 35 mg per dm cube. Answer C. Substrate concentration is limiting at concentrations of glucose phosphate 25 mg per dm cube or less. That is correct, as initially substrate concentration is limiting, and since they are talking about 25 mg per dm cube or less, that means towards the start. Hence the answer is C. If you look at answer D, enzyme concentration is limiting at concentrations of glucose phosphate 25 mg per dm cube or less. That is wrong, as enzyme concentration becomes limiting towards the end and not towards the start. Therefore, based on the results of this investigation, the answer is C. Question 14. What is the definition of the Michaelis-Menten constant Km for an enzyme? The definition for Km is that it is the substrate concentration at which half of the Vmax takes place, and Vmax is the maximum rate of reaction. Therefore, if you go to the answers A, B, C and D, the answer is D, the substrate concentration that gives half of Vmax. Question 15. The graph shows how the rate of entry of glucose into a cell changes as the concentration of glucose outside the cell changes. What is the cause of the plateau at X? A. All the carrier proteins are saturated with glucose. B. The carrier proteins are denatured and no longer able to function. C. The cell has used up its supply of ATP. D. The concentration of glucose inside and outside the cell are equal. Now, y-axis is of the rate of entry of glucose, hence concentrations of glucose inside and outside the cell cannot be equal at X. But if the line was at 0, then it could satisfy option D. Therefore, the answer D is incorrect. C. The cell has used up its supply of ATP. No, that doesn't cause the plateau at X, as the rate of entry of glucose into a cell doesn't use ATP. B. The carrier proteins are denatured and no longer able to function. There cannot be any denaturation taking place, as there are no change in conditions such as pH or temperature. A. All the carrier proteins are saturated with glucose. Yes, that could cause the plateau at X, because if the carrier proteins are saturated, then there are no available carrier proteins to transport the glucose molecules into the cell. Therefore, the answer is A. Question 16. The diagram shows a movement of substance Z across the cell surface membrane. Which process is involved in this movement? As you can see, over time, substance in vesicle moves out of the cell, from inside the cell to outside. Therefore, it should be exocytosis. If you go to the answers, only answer B talks about exocytosis. All the other answers are a form of endocytosis. Hence, the answer is B. Question 17. Whisking tubing is often used as a model during experiments to investigate osmosis in plants. What could whisking tubing be used to represent? Osmosis is when water moves from a higher water potential to a lower water potential, that is down a water potential gradient through a partially permeable or selectively permeable membrane. Hence, since cell wall is freely permeable in nature, it cannot be used to represent whisking tubing since they are investigating osmosis in plants. Therefore, there should be a cross in cell wall, and cell surface membrane and tonoplast are both selectively permeable membranes. Hence, there should be a tick in these columns. And that leaves us with the answer being B. Question 18. A scientist stains the chromosomes of a diploid plant cell with a fluorescent dye to observe the telomeres. This cell has 38 chromosomes. How many telomeres will the scientist observe in one of the nuclei during telophase of mitosis? During telophase, each newly formed nucleus will contain the diploid number of chromosomes, that is 38. Therefore, since each chromosome has one telomere at each end, there will be 38 times 2 telomeres in one nucleus, as each chromosome has one telomere at each end, giving totally 2 per chromosome. And since we have 38 chromosomes, 38 times 2 will give you the number of telomeres in one nucleide during telophase. Therefore, the answer is B, 76, as 38 times 2 is 76. Question 19. The photomicrograph shows a cell during mitosis. What is happening in this cell? Centrioles are replicating, spindle microtubules are shortening, chromatin is condensing. The photomicrograph given shows the process of anaphase, and in anaphase the centromeres separate and the spindle fibers shorten and pull towards each pole of the cell. Therefore, spindle microtubules are shortening is correct and the other options are incorrect. Centrioles do not replicate during anaphase and chromatin doesn't condense during anaphase, it condenses only in prophase. Therefore, the answer is C, 2 only. Question 20. Which events listed are part of mitosis? 1. Interphase, 2. Prophase, 3. Cytokinesis. Mitosis consists of the following stages: prophase, metaphase, anaphase, and telophase. And it doesn't have interphase. As well as cytokinesis is a process which happens after mitosis in the cell cycle. Therefore, the only answer is D, 2 only, as prophase is a part of mitosis. Question 21. The graph shows the length of the spindle fibers during mitosis. Which region of the graph shows when all the centromeres have detached from the spindle fibers? When all centromeres have detached from the spindle, the process of anaphase is complete. Hence, the length of spindle fibers will reach 0. As during anaphase, the spindle fibers shorten in order to separate the centromeres, and once the anaphase process is complete, the spindle fibers will reach length of 0, and that is represented by letter D. Question 22. A short piece of DNA, 19 base pairs long, was analyzed to find the number of nucleotide bases in each of the polynucleotide strands. Some of the results are shown below. How many nucleotide bases containing C were present in strand 1? In DNA, there is complementary base pairing between the two strands. Hence, the percentage of adenine in one strand will equal the percentage of thymine in the other strand. Similarly, the percentage of cytosine in one strand will equal the percentage of guanine in the other strand. Therefore, if strand 1 has 4 of thymine, then strand 2 should have 4 of adenine. And if strand 2 has 7 of cytosine, then strand 1 should have 7 of guanine, since A binds with T and C binds with G. As the question asks how many nucleotide bases containing C were present in strand 1, if we do the math, we get the answer as 3. Therefore, the answer is B. Question 23. Which nucleic acid bases are purines? Now, GAPUR is an easy way to remember this. Basically, it stands for guanine and adenine are purines. And since the question asks us about which bases are purines, that is guanine and adenine. Therefore, the answer is C. Question 24. In a genetic engineering experiment, a piece of double-stranded DNA containing 6,000 nucleotides coding for a specific polypeptide is transcribed and translated. What is the total number of amino acids in this polypeptide? Transcription occurs only on one strand and not on both the strands. And since they are talking about double-stranded DNA containing 6,000 nucleotides, in order to get one strand, we have to divide the total by 2. Therefore, that leaves us with 3,000 nucleotides on one strand. And if we remember the process of transcription and translation, 3 nucleotides forms 1 amino acid. And since they are asking the total number of amino acids, 3000 divided by 3 will give you 1000. Hence, the answer is B, 1000 amino acids in this polypeptide. Question 25. Which statements about tRNA are correct? 1. Hydrogen bonds between bases temporarily hold tRNA against mRNA. 2. The base sequences in the tRNA molecules are the same as the base sequences in the mRNA that is being translated. 3. tRNA translates the base sequence in mRNA into the amino acid sequence in a protein. Obviously, statement 2 is incorrect, as the base sequence in tRNA is not the same as the base sequence in mRNA, but rather it is the same as the base sequence of DNA, except that U becomes T in DNA, as the bases in DNA are adenine, thymine, guanine, and cytosine, and the bases in RNA are adenine, uracil, cytosine, and guanine. Basically, T is replaced by U in RNA. Therefore, statement 2 is obviously incorrect, and that leaves us with answer C being the correct choice, as all the other options consist statement 2. Question 26. The photomicrograph shows a transverse section of part of a dicotyledonous leaf. What are the correct labels for 1, 2, 3 and 4? I have labeled over here what 1, 2, 3 and 4 represents. 1 represents the xylem, 2 represents the phloem, 4 represents the palisade mesophyll since it lies above 3, and the above layer is basically the palisade mesophyll and the bottom layer the spongy mesophyll. Therefore, the answer is D, 1 being the xylem, 2 being the phloem, 3 being the spongy mesophil, and 4 being the palisade mesophil. Question 27. Some of the features present in transport tissues in plants are listed. 1. Lignified walls, 2. Cytoplasm, 3. Mitochondria, 4. Pits, 5. Plasmodesmata. Which of these features are present in phloem sieve tube elements? If you recall correctly, only xylem has lignified walls and pits. And since the question mentions about phloem sieve tube elements, it shouldn't have 1 as well as 4. And that leaves us with the answer being C, 2, 3 and 5. Question 28. The diagram shows the relationship between phloem sieve tube elements, xylem vessel elements and companion cells. Which row is correct? They have shown the diagram, 2 being the similarity between 1 and 3, 4 being the similarity between 3 and 5. Therefore, if we go to the answers A, B, C and D, let's look at A. Companion cells do have endoplasmic reticulum as well as phloem sieve tube elements also has endoplasmic reticulum. Therefore, that is correct. Phloem sieve tube elements as well as xylem vessel elements, they both have no nucleus. Hence, the answer is A. If you look at answer B, companion cells, yes, it has a nucleus. Phloem sieve tube elements, no, it doesn't have a nucleus. Therefore, answer B is incorrect. If we go to answer C, phloem sieve tube elements, mitochondria, yes. Companion cells, mitochondria, yes. Companion cells having a nucleus, yes, that is correct. Xylem vessel elements having a nucleus, no, that is wrong. Therefore, answer C is also incorrect. If you look at D, xylem vessel elements, no cytoplasm, yes, that is correct. Phloem sieve tube elements, no cytoplasm, that is an incorrect statement as phloem sieve tube elements do have a thin layer of cytoplasm. Therefore, the answer is A. Question 29. Which statement correctly describes the transport pathway in plants? A. In the apoplast pathway, water may move through plasmodesmata. B. In the symplast pathway, water may move through intercellular spaces. C. The apoplast pathway may be blocked by the Casparian strip. D. The symplast pathway may be blocked by the tonoplast. Plasmodesmata is only involved in the symplast pathway. The apoplast pathway involves movement through cell walls as well as intercellular air spaces. Therefore, answer A and B are both incorrect. If we go to answer C and D, C. The apoplast pathway may be blocked by the Casparian strip. Yes, that is correct, as apoplast pathway is blocked in the endodermis due to presence of Casparian strip. Hence, the answer is C. Why is answer D wrong? The symplast pathway may be blocked by the tonoplast. Well, tonoplast doesn't block the symplast pathway, but rather allows the symplast pathway to occur due to osmosis through tonoplast. Therefore, D is incorrect. Question 30. The photograph shows an external view of the front of a mammalian heart. Which row identifies the position of the structures labeled P, Q, R and S? Now, obviously Q lies to the bottom right of the heart. Therefore, it is of a ventricle. And since it is to the bottom right, it is the left ventricle. And P represents the left atrium since it is above Q, however, to the right side. Right side of the heart represents the left atrium and left ventricle. Left side of the heart represents the right atrium and right ventricle. Since P lies on the right side of the heart, it is the left atrium. Therefore, giving us the answer being either A or B. And if you look at S, S cannot be pulmonary vein as the pulmonary vein brings oxygenated blood to the left side of the heart. However, S over here represents the right side of the heart. Hence, A is incorrect, leaving us with the answer being B. P being the left atrium, Q being the left ventricle, R being the coronary artery, and S being the vena cava. Question 31. What happens during ventricular systole? Now I have underlined over here the key part of this question. Remember, they are talking about what happens during, not what leads to ventricular systole. If we go to the statements 1, 2 and 3, statement 1, the atrioventricular node transmits an electrical signal to the apex of the heart. That statement leads to ventricular systole and doesn't occur during ventricular systole. Therefore, the answers with statement 1 is incorrect, leaving us with the answer being C or D. If we go to statement 2, the pressure in the ventricles drops below the pressure in the atria, that is incorrect because during ventricular systole, the pressure in the ventricles increases as it forces blood out of the ventricles to the pulmonary artery or aorta. therefore leaving us with the answer being 3 only. That is option D. Question 32. Athletes often move from low altitude to high altitude to train for a race. Which statements about the effect of training at high altitude are correct? 1. Higher concentrations of carbon dioxide stimulate greater oxygen dissociation. 2. Lower concentrations of oxygen stimulate the uptake of more oxygen by red blood cells. 3. Lower partial pressures of oxygen stimulate higher production of red blood cells. If a person moves from low altitude to high altitude, the partial pressure of oxygen decreases because at high altitude there is low oxygen levels. When there is low oxygen levels, the athlete produces more red blood cells in order to compensate for the reduced partial pressure of oxygen. Therefore, statement 3 is correct. Statement 2 is obviously incorrect, as lower concentration leads to production of more red blood cells only to compensate for the reduced partial pressure of oxygen. It doesn't stimulate the uptake of more oxygen by red blood cells, but rather it increases the red blood cell count. Therefore, statement 2 is obviously incorrect. Statement 1. High concentrations of carbon dioxide stimulate great oxygen dissociation. The athletes train at high altitude in order to get more oxygen delivered to the body, hence more red blood cells instead of oxygen dissociation. Therefore, the answer should be 3 only, that is D. Question 33. The graph shows the oxygen hemoglobin dissociation curves at pH 7.6 and at pH 7.2. Which point on the graph shows the percentage saturation of hemoglobin in the blood, leaving an active muscle? Active muscle will have lower percentage saturation of hemoglobin and lower pH due to increased partial pressure of carbon dioxide caused by respiration. Because when a muscle is actively respiring, oxygen levels are used up and carbon dioxide concentrations increases. Therefore, higher the carbon dioxide levels, more combine with water in the presence of carbonic anhydrase, which is the enzyme, forming carbonic acid. Hence, lower pH. Therefore, if we go through the answers A, B, C and D, answer A and C is incorrect because they have a higher percentage saturation of hemoglobin, whereas in an active muscle, they should have lower percentage saturation of hemoglobin at the same partial pressures, leaving us with the answer being either B or D. Why is D incorrect? Because D has a higher partial pressure of oxygen than at B, whereas in muscles have lower partial pressure of oxygen. Therefore, the answer is B. Question 34. Which row correctly describes the short-term effects of carbon monoxide and nicotine on the body of a smoker? The columns are demand for oxygen and concentration of oxygen in blood. The key thing you should know about over here is that nicotine affects heart rate, blood pressure as well as the demand for oxygen, whereas carbon monoxide affects the concentration of oxygen in blood as it binds with hemoglobin to form carboxyhemoglobin. Therefore, if we go to the answers A, B, C and D, A. Demand for oxygen decreased by carbon monoxide. That is incorrect, as carbon monoxide doesn't decrease the demand for oxygen but rather the concentration of oxygen in blood. Hence, answer is incorrect. Answer B. Demand for oxygen decreased by nicotine. Yes, that is correct. Concentration of oxygen in blood increased by carbon monoxide. That is incorrect because when there is presence of carbon monoxide, the concentration of oxygen reduces. Therefore, there is a decreased effect. Hence, answer B is incorrect. Answer C. Demand for oxygen increased by carbon monoxide. Again, that is incorrect as carbon monoxide doesn't have anything to do with demand for oxygen. And that leaves us with the final answer being D. Demand for oxygen increased by nicotine. Concentration of oxygen in blood decreased by carbon monoxide. Question 35. Which statements about bronchioles are correct? 1. They have ciliated cells. 2. They have goblet cells. 3. They have muscle tissue. Bronchioles do not have goblet cells. Therefore, the answers having statement 2 is obviously incorrect, leaving us with the answer being C. 1 and 3 only. Question 36. Which features are important for the diffusion of oxygen out of an alveolus? 1. Blood pressure forces red blood cells through capillaries. 2. Epithelium is permeable to respiratory gases. 3. Moist squamous epithelium present. 4. Surfactant present to reduce surface tension. 2 and 3 are very important features for the diffusion of oxygen out of an alveolus. Hence, the answer which has both 2 and 3 is only A. Therefore, A is the correct choice. Question 37. The diagram shows some of the pathogens that cause disease in humans and some of the ways they are transmitted. What is the correct pathogen and method of transmission for measles? Measles is caused by a virus. Therefore, if you go to the pathogens, it cannot be bacterium and it is definitely not a Productis. That leaves us with the answer being 3, morbillivirus. Hence, we can rule out answers A and B. Measles is airborne due to it being an aerosol or droplet infection. Therefore, the answer is X, coughs and sneezes. Hence, the answer is D, 3 and X. Question 38. Which row is correct for each disease? The columns are cholera, HIV, AIDS, malaria as well as measles. If we go to the answers A, B, C and D, A, caused by bacterium, yes, that is true for cholera and the bacterium is called Vibrio cholerae. HIV AIDS can be transmitted in breast milk and across placenta, yes, that is also true. Malaria eradication program is unsuccessful, yes, that is also correct. Measles caused by a virus, that is also correct, and that virus is called morbillivirus. Hence the answer is A. If we look at the other answers B, C and D, in B they have mentioned, under malaria they have mentioned carried by male Anopheles mosquitoes. That is incorrect, as male mosquitoes has nothing to do with malaria. It is the female mosquitoes which act as the vector. Therefore answer B is incorrect. If you look at answer C, cholera airborne infection, that is incorrect, as cholera is a waterborne infection. If you look at answer D, under cholera can be caught by swimming in contaminated water, that is incorrect, because cholera can only be caught if you drink contaminated water and not swim in it. Therefore answer D is obviously incorrect. Answer A is the correct choice. Question 39. Which statements describe myasthenia gravis? Myasthenia gravis is a condition where the body fails to distinguish between self and non-self antigens and therefore attacks the proteins within the body. Therefore, statement 1 is obviously correct. Antibodies attack proteins within the body. Yes, that is true. If you look at statement 2, T lymphocytes are involved in an inflammatory response. Now, what do you mean by inflammatory response? That response occurs towards pathogens, hence it is not related to myasthenia gravis, as myasthenia gravis attacks body cells and not pathogens. Therefore, statement 2 doesn't describe myasthenia gravis. Statement 3. The immune system blocks receptors at the neuromuscular junction. Yes, that is correct. That is why it leads to muscle weakness, as the muscle cannot be repeatedly stimulated due to blockage of the receptors at the neuromuscular junction. Therefore, only 2 statements can be true. And since 1 and 3 are definitely correct and they describe myasthenia gravis, the answer is A, 1 and 3. Question 40. Which row shows the cells that are able to divide continuously and are involved in monoclonal antibody production? Cancer cells, mouse B lymphocyte plasma cells, and hybridoma cells. Actually, all 3 are involved in monoclonal antibody production. However, when you consider the ability to divide continuously, plasma cells are only able to produce antibodies and cannot undergo multiple cell divisions. Therefore, the correct answer is C. Cancer cells are able to divide continuously and are involved in monoclonal antibody production. B lymphocyte plasma cells are unable to divide continuously, hence that is why hybridoma cells are obtained by fusing them with cancer cells to give the hybridoma cell the ability to divide multiple times and produce the required antibody. Therefore, the answer is C. And that is the end of the paper. If you made it this far, thank you for sticking by. Appreciate it more if you could possibly share with your friends too. Let's keep the biosquad growing and stay tuned for more.